Okay, so welcome to our first quantitative and computational biology seminar series talk of this academic session. Normally, this is a more local seminar series that we ran in Montreal to have kind of crosstalk between experimentalists and quantitative researchers who could work at the interface of these two kind of fields and to increase like collaborative possibilities. And so I'm really in some way not happy that there's pandemic, but it has worked out well for us because now we get speakers from all over the place. And I'm really excited that Elsje Pienaar, hopefully is okay, is our first speaker. Elsje and I have met earlier actually because of COVID and are working together on some separate projects from her HIV talk, but I'm really excited to hear about her work informing treatment regimen designs of immunotherapy for HIV. So Elsje is an assistant professor in the Weldon School of Biomedical Engineering at Purdue University. She earned her MS and PhD in chemical and biomolecular engineering from the University of Nebraska-Lincoln, and did postdoctoral work in microbiology, immunology, and chemical engineering at the University of Michigan, as well as in Linköping University, Sweden. Her laboratory uses computational simulations of within-host pathogen, immune, and drug dynamics to optimize treatment of infectious diseases. Current projects in the lab include TB, HIV, non-tuberculosis mycobacterial infections, and Ebola virus dynamics, and as I mentioned, some SARS-CoV-2 work. So with that, I will hand it over to Elsje. If you have questions, please use the chat, and we will have a period of questions, hopefully at the end. Thanks.
All right, thank you, Morgan. I'm really excited to be here. I tried to tailor the talk to be considerate of both the biologists and computational people, but if there's anything that's unclear, please just post your questions and I'm happy to clarify towards the end. So as Morgan mentioned, I use systems biology and systems pharmacology methods to look at infectious diseases, their interaction with the host immune system, as well as how we can use these models to design new drugs and optimize drug regimens. And so today, the story that I'm going to tell you is how we do this for HIV and specifically for immunotherapies in HIV. I am going to turn my video off. I don't have a ton of faith in my Wi-Fi here, so I would rather you see my slides than my video, but I'll turn it back on for questions later. So hopefully everybody can still hear me. So just quickly, I always like to clarify what I mean by systems biology and systems pharmacology, because oftentimes in these computational fields, systems biology means different things to different people. So what I mean by systems biology and systems pharmacology in my lab is that we look at experimental data that spans interactions from host immunity, immune responses on their own, or host immune interactions with pathogens. And then we also look at experimental data that look at host immunity, how it interacts with drug dynamics, and how the drugs interact with these pathogens. And so there's a whole wide variety of experimental data that look at all of these different combinations and aspects of this interaction. And so our approach is to take all of these different bits and pieces of information and knowledge that we have and put it all into one computational platform. So for example, if we know that a pathogen interacts with macrophages in one way, and we know that it interacts with CD4 T cells in a different way, we can put those interactions together in one computational system and say, well, if both of those things are true, what impact does it have on both the macrophage and the CD4 positive T cell population at the tissue scale? So we built these computational systems that are like virtual versions of the infections that we look at. And these systems in my lab are mostly focused within host. So we don't look at epidemiological models. We look at what's happening inside the body between the virus and your cells and the drugs. So that's kind of a very high level view. Now let's talk about HIV. So I think, as many of us know, HIV is a virus that disproportionately affects the developing world. But this doesn't mean that it's not a problem in the developed world as well. So overall, the highest percentages of HIV prevalence is in sub-Saharan Africa, including my home country of South Africa, which has pretty high numbers. But even in the US, there's 1.2 million people living with HIV currently. Globally, there's 38 million people living with HIV, and about 1.7 million new cases were reported in 2019. And about 700,000 deaths were reported in 2019. So this is still a very important and very critical global public health crisis. And so this is kind of the global views, just to convince you that it's an important problem in the context of all of the other viruses that we have to deal with currently. And so now let's talk about the virus and the infection a little bit more. So HIV, or human immunodeficiency virus, is a retrovirus, and that means that it integrates its genetic material into the cell that it infects. And so it fundamentally alters the cell that it infects. It's a systemic infection, so it can be found in many different organs and tissues, including in the blood. And it mainly targets CD4 positive T cells, although it has been shown to affect other cell types as well. When you get infected, it's initially associated with flu-like symptoms. And this graph on the bottom right here kind of shows that the typical figure that you always see associated with HIV infection, where upon initial infection, you see an early spike in the viral load. And this is associated with a decrease in the CD4 positive T cell count. But then this viral load kind of drops down to some set point. And this set point can be contained for or maintained for months or years, as there is a slow decline in the CD4 positive T cells and a slow creep in the viral load. And then eventually, many years later, the viral load can expand significantly. When the CD4 T cells drop precipitously, that's when patients develop AIDS. And that's associated with fevers, weight loss, and recurrent infections, because the immune system is just so compromised at that point. HIV is treatable, but it's not curable at the moment. Antiretroviral therapy, or ART, can maintain viral loads below detectable limits and can restore CD4 positive T cells, but it's a lifelong treatment. If you remove the treatment, the viral load rebounds in most patients. So that's kind of the infection progression in general. But if we're going to talk about immunotherapies, then let's also talk about some specifics about the immune response to HIV. So this is obviously a very simplified view of the immune response to HIV, and I apologize to immunologists everywhere, but I wanted to keep it super simple so that we could talk about the things that we're going to focus on in our model today. So after you get infected with a virus, the virus will infect healthy cells, mainly CD4 positive T cells. When these cells become infected, as I said, the virus integrates its genetic material and kind of co-opts the cell to produce more viruses. These viruses then go on to infect more cells. And so this cycle is kind of what sustains the infection. These cells, once they're infected, of course, they can die from the infection or just from other cell death events. And then on the immune side, there's cytotoxic cells, and the figure here, I just call them killer cells, but they represent CD8 positive T cells, so cytotoxic T cells, which are adaptive immune cells, and they're antigen specific. And there's a separate population of cells called natural killer cells, which are also cytotoxic, but they are not antigen specific. And both of these cells can interact with infected cells and kill them. So this is in a very, very simplified way. One of the immune responses to this infection is to have these cytotoxic T cells that will come and kill the infected cells and then prevent them from making more virus. And so if we're talking about antiretrovirals, ARTs, what they do is they target various different viral proteins, and they essentially interrupt this infection process, either by inhibiting the production of new viruses or inhibiting the binding of the virus to healthy cells. And so it kind of cuts off this viral production cycle. So at this point, you might say that if we have ART and it works as long as people take it, then what's the problem? But as always, things are not quite as simple as they might seem. So let's talk about some of the complexities around HIV infection and treatment. So even though ARTs work really well, only about 67% of people living with HIV actually have access to them. And this question of ART access is a very complex research field all on its own. That's not important here today, but you can imagine that If we were able to design a therapy that needed fewer doses, that you don't need to take drugs every single day, or if you don't have to take it for the rest of your life, that a lot of these cost and infrastructure hurdles to treatment access would become lower so that we can increase access to more people. Beyond kind of the infrastructure questions, just having to take drugs every day for the rest of your life puts a huge burden on people, on the patients. You can imagine that if you have to take something for the rest of your life, that it starts to kind of weigh on you. And it also creates compliance problems. And if patients are non-compliant, if they skip doses, or if they fall off treatment for a few months, this really can lead to drug resistance and viral rebound. And so again, if there's treatment with fewer doses or shorter treatment, it would really help this burden on the patients as well as the compliance questions. And then talking a little bit more specifically about the pathology of the disease. So this graph that I showed you earlier really focuses on the CD4 T cell counts. And even though ARTs can restore CD4 T cell counts, those numbers go back up. Even on ART, there is chronic inflammation that persists in patients living with HIV. And this chronic inflammation has been associated with lots of different comorbidities, including cardiovascular disease, neurocognitive impairment, and just lots of other complications. And so even though the viral loads are down, the CD4 T cells go back up, your body isn't quite back to quote unquote normal. And then finally, also related to the CD4 T cell level, it has also been found that even patients on ART with restored CD4 T cell levels, the CD8 T cell numbers remain elevated because of this continuous chronic exposure to viral antigens. So even on ART, if the viral load is controlled, the patient's system is not quite at normal levels, and it has all of these other comorbidities associated to it. So I'm telling you all of this to at least make the points that even though ART can suppress viral replication, we still need to come up with better alternatives or improvements to ARTs or complementary treatments to ARTs to improve just outcomes and patient quality of life for these patients. So if we talked about antiretroviral therapy, kind of cutting off the faucet here of making new viruses, what immunotherapy does is it promotes the immune system's own responses to killing infected cells. So kind of pulling the plug on the infection as well as turning off the tap, the faucet on the infection. So if there's a way for us to supplement kind of the natural immune response, maybe we can get a better handle on the virus. And so one type of immunotherapy that I'm going to focus on today is centered around IL-15. And this is in collaboration with Dr. Shelby O'Connor at the University of Wisconsin in Madison. So IL-15 is a cytokine that stimulates both the proliferation of these cytotoxic cells as well as their ability to kill infected cells. So IL-15 is a molecule that the body makes naturally, and it has these immune stimulatory functions. And so this should be a good immunotherapy, right? Only the problem is if you just inject IL-15 into patients, it doesn't really do a very good job as a drug. And so people have developed this IL-15 agonist. So it's an engineered molecule that mimics IL-15, but it's more effective and it has a better pharmacokinetic property. So it hangs around in the body longer. And so what this molecule is it's made up of a mutant IL-15, the N72D mutant, that has a better binding affinity to the IL-15 receptor. And then it's fused with an IL-15 receptor alpha subunit domain, as well as an IgG Fc domain. And so this drug is called N803. It was previously called Alt803, so you might see both of those if you're looking in the literature. And so what this molecule does is it has the same types of functions of IL-15, but it's more drug-like. So it stimulates the IL-15 pathway so that CD8 positive T cells and natural killer cells, NK cells, proliferate more and increase their cytotoxic activity. And N803 does this pretty well. So here are some results from cancer trials where N803 was given to cancer patients and then in 4 doses, subcutaneously or IV. And today I'm just going to focus on the subcutaneous one because that's what was done in the data that I use as well. And so you can see that this really has a significant increase in both natural killer NK cells as well as CD8 positive T cells. And this was in patients with leukemia and lymphoma. And they did see a clinical response in about 20% of patients here. And so in cancer, at least, it has been shown that N803 can induce these cytotoxic T cell populations, and that in some cases it can have a clinically significant effect. So if NK cells and CD8 positive T cells are important in controlling HIV, and N803 can stimulate both of these populations, then can we use N803 to reduce viral load in patients with HIV? And so our collaborators in Wisconsin set out to do just that in a non-human primate model of HIV. And so these non-human primates are infected with SIV, which is the primate analog to HIV. And so these monkeys were infected for between 1.5 and 3 years with SIV. So they were chronically infected before they were given this treatment. And then at day 0 here, they were given 4 doses of N803 in the first cycle. And then there was a 2-week break, and then they were given a second cycle of 4 doses. And then there was a long 29-week break, and then they were given a third cycle of 4 doses. And these were subcutaneous doses, like we just discussed in the cancer data. So this is our non-human primate study that we looked at. This is a very small study. There's only 3 monkeys in this study, so that's one of the limitations of this study, but it's unfortunately a kind of a challenge with non-human primate studies in general. So these were the doses that the monkeys received, and so we can look at the fold change in their NK cell responses. And so we'd see in the first cycle, in 2 of the monkeys, a pretty strong response, and then the subsequent cycles, we do see a response for in 2 of the 3 monkeys. And then in the third cycle, can you still see my screen? I feel like it just disappeared for a second.
We can still see it, yeah.
Okay. And then the third cycle, we see a little bit of a reduced response, but it's not quite so pronounced as in the first cycle. And then we can also look at the CD8 positive T cell changes, see kind of a similar response. It's kind of messy. There's a lot of variation between the primates, which is also kind of expected with these animal studies. But we do see a response for every treatment cycle. And then some contraction in these cell numbers between cycles. So the cells kind of go up and then they come back down between cycle 1 and 2, and then they go up and they come back down. And then in cycle 3 again, smaller peaks here, but then they come back down. And so in both of these populations, they were elevated during treatment, and then the stimulatory effect kind of waned. So at least so far, this is consistent with what people saw in the cancer patients. And so now if we look at the viral response in these 3 animals, there are some key qualitative things that I want you to notice, and I'm kind of going to slow down here because we're going to talk about it again later. So what we see in the first cycle is that there's a really strong reduction in viral load. But within this first cycle, while they're still getting doses, you see already a rebound in virus. The virus is regrowing even though they're still receiving treatment. And then in the second treatment cycle, you see almost no response. And then in the third cycle, we see a little bit of a recovery. You see some drop in the virus, but as with the first cycle, while they're still receiving doses, the virus is going back up in some of these. So these are kind of the key qualitative things that I want you to keep in mind about this data set. So this is what we're going to try to build a simulation to reproduce these dynamics. So this transient response in the cell numbers, and then these qualitatively different responses in the viral load between treatment cycles. So now that we have this data, this, as usual, brings up more questions than answers. So these are some central questions around this treatment. So why does the viral load decrease in phase 1, but then in phase 2, we don't see any response? Or why do we recover some efficacy in phase 3? What's special about that? And then we're seeing newly proliferated NK cells and CD8 positive T cells. Are they functional? What are they doing in this system? And then finally, If we see a transient response in this data set, can we design a different regimen where we can maybe get more persistent responses in the viral load? And so I just want to spend a couple of minutes talking about why these questions are difficult to answer. And some of this relates to the reason why our computational models are helpful. So the immune response, our immune systems has evolved to have these very complex built-in feedback loops to inhibit inflammation. So, and these are designed to avoid autoimmune disorders. And they're very complex and they're very integrated and they're there for a reason and they're really, really good, but it makes it difficult to give these immune stimulatory treatments because every time you stimulate the immune system, all of these feedback loops are going to kick in to suppress the response that we're trying to induce. And then the second problem is that these dynamics are quite complex and they're multi-scale. You have things going on from the molecular scale all the way up to the host scale that are interacting with each other. And those things are often very difficult to analyze experimentally. And then finally, kind of related to the multi-scale nature is that all of these things are changing simultaneously. So the drug concentrations and the viral numbers, they're all changing simultaneously and they're interacting with each other. And it's really difficult to kind of pull those things apart and untangle them in experimental data. And so for all of these reasons, the computational models are helpful. So what we did in this case to try to address some of these challenges is to build a very simple ordinary differential equation ODE model of this system. So here's the infection and immune response figure that you already saw. And so we have part of our model that represents the infection, so the interaction between the virus and its target cells. We have another part of our equations that look at the cellular immune response. Again, this very simplified cellular immune response, including separate representation for CD8+ T cells and NK cells. And then we have a different model that looks at the N803 pharmacokinetics and pharmacodynamics, so how the concentration changes over time and how it impacts the cells to stimulate both proliferation and killing. So now that we have this model that kind of represents the system, now we can go back to the biology for a minute to see what hypotheses are there for explaining this N803 response, and can we use our model to test them? So going back to our experimental collaborators' data, they identified 3 possible reasons that could explain the dynamics that they saw. And the first one relates to drug tolerance, and this relates to receptor internalization. So they measured the number of IL-15 receptors on the surface of the CD8+ and NK cells and noticed that the number of receptors on the surface decreased over time with treatment. So if this receptor is the target of the treatment, obviously if the receptors get internalized, there's nothing to target, so the drug won't work. And we term this in our simulation drug tolerance, and it represents the internalization of this receptor. Another mechanism, or kind of a group of mechanisms that they identified, relates to all of those feedback loops that I just talked about. So they noticed an increase in regulatory markers, including PD-1 and CD39 on their cells. And they also noticed an increase in regulatory T cells in these monkeys. And all of these serve to suppress proliferation and activation of effector immune cells. And then in a separate study, people have found that N803 can induce IL-10 expression, which is a cytokine that suppresses pro-inflammatory cytokines. And so that will also contribute to just a general dampening of the immune response. So obviously this is a lot of very complex interactions, but for this preliminary model, we're just lumping them all together in what we term immune regulation. And it's something that downregulates both the proliferation and the killing of the cytotoxicity of the CD8+ and NK cells. So this is a very coarse-grained view, but for this initial analysis, we just wanted to separate kind of the more systemic immune responses from the effects that are drug-specific, so the receptor internalization. And then there's one final mechanism that they found in the paper, is that they saw a change in the amino acid sequences of the viral population. So they see a change in the viral variants that are present in these animals. And what this represents is those variants that are targeted really well by the CD8 T cell populations, those will be depleted, and this will give other viral variants that are maybe not targeted so well a chance to kind of grow a little bit better. And so based on this experimental information, we kind of focus on these three mechanisms, and we call them drug tolerance, immune regulation, and viral escape. So these are the things that we're going to test in our model to see, are these contributing, or can they be contributing to the viral response that we see, and if so, how much? And so I'm going to kind of skip through these next couple of slides very quickly. I'm happy to come back and talk more details if there's someone who has questions about the equations. But just very briefly, we have 4 different ODEs that represent the infection. So you can see that's very simple. We have one for the CD8 positive T cells, one for the NK cells, and then 2 equations, one for each of our viral variants. So we built a model that just has 2 variants in it to see if we can capture this dynamic of the change between the viral variants. And then we have a second set of equations that represent the concentration of N803, both in the injection site and in the plasma. And then these terms that represent the drug effect on both cytotoxic killing and proliferation, and then how drug tolerance and immune regulation impacts the drug effect as well as the general immune response. So again, I'm happy to come back to this for more details in the questions. So if we build this model with all 3 of these mechanisms in it, can we match the data? And I guess I wouldn't be telling the story if the answer was no. So we can reproduce these key features that we talked about earlier in the data. So the experimental data is shown here in gray again, and then the black lines in the black shaded area represents the top 1% of all of our calibrated model parameter sets. So when we calibrate this model, we do a multi-start random search. So we choose a bunch of different initial conditions for our search algorithm and then run through the optimization to try to minimize the distance between our experimental data and our simulations. And then we pick the top 1% results. So we're not making any strong statements about particular parameter values. We're looking at a population of models so that we account for this uncertainty in our very coarse-grained model. So what you're seeing here is kind of a standard deviation on our model predictions. And so you can see that we're able to capture the strong drop in the viral load in the initial cycle, very limited response in the second cycle, and then some recovery of efficacy, but not quite as strong as in cycle 1 in the third cycle. We're also able to capture this response in the cells, the contraction kind of between treatments, as well as some recovery after in the third cycle. Okay, so if we put all of the mechanisms together, we can match the data, but this doesn't get us any closer to understanding what each of these mechanisms are doing. So to dig a little bit deeper, luckily in the model, it's really easy for us to turn some of these things on and off. And so we wanted to look a little bit closer at each one of these mechanisms to see if they are necessary to reproduce the data. So we started with our full model, which is the one that I just showed you. And then we systematically remove these mechanisms. So for example, in model 1, it includes immune regulation and viral escape, but it does not have any drug tolerance built in. And then we calibrate this model independently of the full model and see if it can still reproduce the data. And then we do the same thing for model 2, which doesn't have immune regulation, and model 3, which doesn't have viral escape. So we're systematically looking at each of these mechanisms. And then we're comparing them quantitatively. So we're looking at the negative log likelihood as well as the AIC to see how well they fit the data. And then we're also looking at qualitative criteria where we're asking how well they capture the dynamics in each of these cycle kind of qualitatively. So again, if we have our full model calibration here, here's where those quantitative criteria land, and it is able to produce all of these qualitative features. So now if we look at each of these models independently, model number 1 can capture all of the qualitative behaviors. The quantitative metrics are a little bit worse, but overall the fit's not too bad. If we take out immune regulation in model number 2, you can see that we can essentially just fit a flat line here. So we're really not able to match any of the qualitative criteria. And so this suggests that a model without immune regulation is not really able to reproduce key features of the data. And so that suggests that this immune regulatory mechanism is really, it's necessary to be able to capture the dynamics. And then if we look at the third model that doesn't have viral escape, again, it's able to reproduce the qualitative behaviors, and the quantitative metrics are also quite good. So model 1 and model 3 are both able to capture kind of the key features of the data that we're looking at. And then there's one final model that we did just to say, just to see if immune regulation on its own is able to reproduce the data, since the tolerance and viral escape don't seem to be necessary. But if we remove both tolerance and viral escape, we see that we're able to reproduce kind of the short-term dynamics for each cycle, but the response in cycle 1 and cycle 3 are identical. So you lose this long-term change in drug efficacy if you don't have either of those mechanisms. So if you take both of them out, you can't reproduce the qualitative criteria. And so that means that you at least need one of them, but we're not sure which one or if we need some combination of the two. So what these results suggest is that either a model with immune regulation and drug tolerance or immune regulation and viral escape is able to reproduce the data. And so at this point, we don't really know which one of these are true or if it's some combination of the two. So we're just moving forward with our analysis, kind of taking this uncertainty into account to see if we can make any kind of consistent predictions. So moving forward, I'm going to show you data that looks like this. That's color-coded again for each of the models, but now we're going to focus in on model number 1 and model number 3. So at this point, we know that these two mechanisms, immune regulation and either tolerance or viral escape, is necessary to reproduce the data, but we don't really know how much and when and where and how these mechanisms are contributing to the outcomes that we see. So to try to dig a little bit deeper into this question, we kind of developed this metric called the per cell killing. And this is kind of mathematically equivalent to a per infected cell per effector cell killing rate constant. So we extracted from our ODEs the total killing rate, and then we divided that by the total number of killer cells and the total infected cells. So you get a killing rate per killer cell per infected cell. So it kind of gives you a metric for how good each of your individual effector cells are at killing infected cells. So if we look at this per cell killing metric that I just told you about for model 1, over time through these 3 cycles, we see that very early on, there's a sharp spike in the per cell killing. So the cells that are there get pretty good at killing infected cells, but then very quickly this per cell killing drops. And then in cycle 2, this doesn't really recover. And in cycle 3, you get a little bit of a spike, and then it drops even further. So you can see that the model is predicting that this per cell killing is kind of going down over time. And this would help explain why the viral load is not responding, because the immune cells that are there are not doing a very good job of killing the infected cells. So in this model, remember that we have both viral escape mechanisms as well as immune regulatory mechanisms that are driving this change in the per cell killing. So what we can do with the model is we can pull these apart even more. So we can ask how much of this per cell killing change is due to immune regulation. And what we see here is that it's a very consistent response for each treatment cycle, is that the immune regulation lowers the per cell killing in the first cycle, but then once this treatment ends, the per cell killing kind of goes up quite quickly, and then it goes down again in the next cycle. So it seems to be a very short-term effect. Whereas the per cell killing reduction due to this viral escape is much more long-term, and you can see that these dynamics are really the ones that are driving this long-term change in the overall per cell killing. So this is suggesting that the regulation is responsible for kind of these short-term changes that we see within each cycle, and that the viral escape persists across cycles. And this kind of makes sense because this shift in the viral population is not something that is typically reversible. The viral populations are going to keep evolving as the host immune system changes. So now if we look at model 3 to see what is going on there, we see very similar patterns in the per cell killing, where there's an early spike, a little bit bigger spike here than before, and then very quickly again, it decreases. And if we look a little bit closer in this model, remember we had drug tolerance and immune regulation. Now, if we look at how much immune regulation is contributing to the reduction in per cell killing, The first thing that you will notice is that this range is much wider. So there's a lot more uncertainty in how much regulation is contributing in this particular model. But we see very similar if you just look at the average trend, short-term impacts for regulation. And then if we look at drug tolerance, we actually see patterns that look quite similar to the immune regulation patterns. And this range in effects here is really quite wide, ranging from having no impact at all to having a 10-fold reduction in per cell killing. And so what these wide ranges are telling us is that within this model that has regulation and drug tolerance, that the regulatory mechanisms and tolerance mechanisms are able to compensate for each other. And so our model can't tell the difference between those two, which is important information to know, because now we need to incorporate that into our analyses moving forward. And we're communicating with our experimental collaborators to try to quantify both of these responses a little bit better so that we can figure out which one of these scenarios are true. So now let's go back to our central questions for this treatment. So why does the viral load decrease in phase 1, but not in phase 2? And so what our model suggests is that inhibitory signals, including regulatory markers and either the tolerance or the viral escape, triggered in cycle 1, still persist by the time that cycle 2 starts. So the local conditions are not kind of good for N803 treatment by the time that cycle 2 starts. And so for the second question, why do we recover some efficacy in phase 3? The model suggests that break is long enough to for some of those inhibitory signals to kind of normalize, although not completely. And then the third question, our simulations also suggest that there's a significant reduction in functionality in these cells. And what the model has really identified is that given the proliferation of the cells that has been experimentally measured, there must be some sort of a reduction in functionality to be able to explain the rebound in viral load. And we were able to quantify that using the model. So now let's turn our attention to the final question. Based on what we've seen so far, it suggests that the efficacy of this drug really depends on the timing of the doses relative to the timing of these various host responses to this stimulatory drug. So this observation kind of suggests the question that if we space the doses out a little bit more to give more time for the immune response to recover or to normalize between doses, can we improve treatment outcomes? And so this is something that we can quite easily do in our simulations. And so if we do that here, we did the weekly doses, or we space the doses out 2, 3, or 4 weeks apart from each other. And you see that in model number 1 that has viral escape and immune regulation, we do see a statistically significant, but not a very strong reduction in the overall viral load at the end of treatment. So it does suggest that dose spacing can help in this case. But if we look at model 3, we see that there's a possibility for a much stronger reduction in viral load. And so what this suggests is that depending on what the underlying mechanism is, if it's viral escape or drug tolerance or some combination of the two, that driving mechanism is going to determine the impact of dose spacing. And it also gives us a potential testable prediction that if the viral escape is mainly driving outcomes in the monkeys, then if we go back and do another treatment cycle with monkeys and we don't really see a change with spacing out the doses, then maybe it suggests that the viral escape is a bigger driver of outcomes than drug tolerance would be. And so there's one more treatment change that we can talk about, is if our model suggests that immune regulation is such a strong driver of why N803 stops working after some time, then we can ask if there was a way that we can block this regulatory response. Can we improve the treatment outcomes? And so remember that one of the regulatory mechanisms that we included one of these regulatory markers is PD-1, and there are very effective cancer drugs available that target PD-1, PD-L1 pathways. So this is a potential thing that we could do, is target some of these regulatory markers to try to inhibit this regulatory signal and hopefully give N803 a leg up so that it can keep being more effective for longer. So we can do this in a very phenomenological way by just progressively decreasing by a certain percentage the strength of this immune regulatory mechanism in our model. And so if we do this in model number 1 that has the viral escape, we see that there is quite a large reduction in the overall viral load at the end of week 15. And as with the dose spacing, we see that there's a stronger decrease in the drug tolerance model. So in both models, it suggests that this approach, this combination therapy approach, it could be a good strategy to complement N803 treatment. And so it also indicates that some combination immunotherapies could help. So going back to our central questions, what other approaches might work? Our model suggests that longer dosing periods between doses, or very targeted, and I do stress targeted, combination immunotherapies could work, but that the impact of these approaches is going to depend on what the underlying mechanism driving the dynamics is. And that at this point is not clear. It needs more information. So as a quick summary, I've told you today about how we use our systems pharmacology approaches to quantify potential mechanisms driving loss of efficacy for this drug, and also how we can use it to develop new treatments. Of course, we need to talk about limitations. This is a very, very simplified model, very phenomenological model. We don't include things like latent viral reservoirs. We haven't talked about dynamics in lymphoid tissues or other tissues. We haven't talked about antibody responses. So it by no means is a comprehensive model, but our model is able to parse out the potential contributions of these wider categories of mechanisms that kind of incorporate some of these specifics that I just mentioned. There's also limitations in our data. As I mentioned, this is just one data set, and this is a very specific set of monkeys with a very specific genetic background and vaccination history and infection history. And so all of our conclusions are very much limited to this data set. But the nature of our computational approaches is that we are able to integrate multiple data sets. So our collaborators are currently collecting another data set with this drug that we can then incorporate into our analysis to make the model even more generalizable. And I mentioned the small sample size earlier, but it's kind of a normal problem in these primate studies. And then finally, regarding the next steps, the story that I've told you today is really only the first half of the iterative loop between experiments and simulations, the simulations making predictions. And now these predictions really need to be confirmed in primate studies, and we are working closely with our collaborators to come up with the best way to do that and figure out exactly what things we need to measure so that we can answer some of the questions that we raised here. So things like, can we further clarify how much models 1 versus model 3, the tolerance versus the viral escape, is contributing to this infection outcome. So that's the end of my story. I would like to thank my lab at Purdue. Jonathan Cody is the student who did all of this great work. Our collaborators in Wisconsin, who have been very generous with their time and their expertise and their data, as well as these funding sources that have supported our work. And then I am happy to take any questions.
Thank you, Elsje. That was a beautiful study. I have some questions that I can hold off. If there is anybody who would like to ask a question, you could write in the chat, and you can, I'll invite you to maybe unmute yourself and you can ask it orally instead of textually. But maybe I'll give you a moment to ask a question. I was actually really, before you went into the anti-PD-1, PD-L1 blockade, that was my big question. So stole it, but that's probably good because I think that that's important. So this suggests, because a lot of research in cancer immunotherapy is thinking about combination strategies, particularly with ICIs that have shown like a lot of efficacy in certain cancer subtypes, but less in others. So I think that that's a particularly interesting result that potentially could have implications in in cancer therapy. So I don't know if you have some thoughts about whether this could be translatable. I mean, obviously the mechanisms in your model are going to change, but the immune regulation mechanisms could be transferred over to different oncological applications.
Right, right. And I think this is something that we've talked about for the SARS-CoV-2 project too, is that your immune system doesn't change. It's just whatever it's being stimulated with that changes. So yes, I do hope that if we can build a very detailed model of N803, how it interacts with the IL-15 pathway, all of these downstream things that get affected by stimulating this IL-15 pathway, that could be a kind of transferred to other models where similar cell types and similar molecules are involved. And then the other thing that I think is interesting that we haven't touched on here because our sample size is too small is what the initial conditions is when you give the treatment, because our changes over time really suggests that what the current state of the immune system is really determines whether this drug is going to do well or not. And then it kind of raises the interesting question of, is it possible for us to name that? So can we identify good initial conditions? And then is there a way to identify that in certain patients? Because I don't know a ton about the cancer responses, but the little bit that I've read does indicate that you usually have like a subsection of patients that respond to this treatment. And so if there's a way for us to figure out what is it about those patients, then can we either target them with the drug so that we don't waste it on other people or waste other people's time when we're trying to treat it? Or can we try to recreate those initial conditions with combination therapies or other sorts of interventions?
Yeah, there's definitely like a stratification of the responses, like you said. And I also think it's interesting you talk about kind of modular design, because I think a lot of what we're all doing is kind of building these little modules that then could be used in other places. So that's very interesting. Alan Perlson has a few questions that he would like to orally. So Alan, why don't you, I think you can unmute yourself and turn the camera on if you want.
Yeah. Can you hear me? Hi. So that was very lovely talk. So I do have some questions. One, I'm a great believer in phenomenological models, so I sort of like that approach. But there's a huge amount of data on SIV infection in non-human primates. Have you tried to take your phenomenological model without any drug and see if you can fit data, historical data? I'm a little bit skeptical that your model will do that, but it'd be nice to know if you've tried and what the results are.
Yeah, so your skepticism is well placed. It's really hard for us to do. And so that's why we've kind of focused on this treatment phase. So what we've had to do, because we have tried, because these monkeys that we used actually were part of separate vaccine trials. And so I think for 2 of them, we actually have acute infection data. And we did try, but the model just couldn't do it. And so for now, we've kind of assumed that we're building our model to represent the chronic stage of infection. But honestly, I think that limitation is part of the problem for why we're having some identifiability issues, and really trying to build a model that can capture the whole infection cycle, I think will get us a little bit closer to be able to say something more definitive.
Yeah, yeah, we've had better success doing that, fitting the acute infection part first and getting some parameters that describe the infection in individual animals. Second, just to comment on the model, I don't know if this was a typo or if it's correct as you showed the equations for the CD8 response or the effector cell response. It wasn't driven by anything. I mean, it was just totally autonomous, both the NK and the effector cell response. It wasn't responding to virus or infected cells. They didn't play a role in those equations at all.
Right. And that is a side effect of our chronic infection assumption, that the baseline infection and viral contributions are kind of rolled into the background parameters. And so the only thing that's new in kind of this treatment phase is this drug concentrations driving both the cytotoxic activity and the proliferation.
Right, but CD8s need to be stimulated, not only by the drug, but by seeing infected cells, for example, that they have to interact with to drive that. So I have some concerns. The other thing that you don't have any limitation on target cells, I presume you're just holding them constant here. So even when the virus loads get high, there's no feedback that they actually kill those target cells. So there's less target cells available which may be part of the way, the reason that the virus comes down. At least that's what happens in acute infection.
Right, right. And that was a very deliberate decision that we had made here, that we're kind of saying that the infected cells follow the viral load quite closely. And we kind of had to do that again, just for identifiability issues, because we didn't have measurements of those two metrics separately. So the number of infected cells and the viral load separately. But yeah, I agree that there is a limitation. What we're hoping here is that since we're in the chronic phase of the infection, that there won't be these massive spikes in viral load that will really drive down the target cells too much. But of course, without actually doing it, we can't say for sure.
And then just some quick things on the biology. Since you have viral escape, has the virus been sequenced? I mean, you should be able to tell in individual animals if there is escape or not. It's not, you don't have to do it with your model.
Right. And so the reason why we built it, sorry.
So is there a viral escape? And it should be different for different animals. Not every animal will respond the same way. So I would be fitting the animals independently, especially if you think viral escape is important. But anyway, the first question is there evidence of viral escape, and what sort of kinetics? Is it present in the first cycle, second, third, or?
So there is evidence of viral escape, and that's the reason we put it in the model, because that was one of the hypotheses that came out of the biology. And so they did sequence the viruses, but we don't have any sort of time course for it. We have a before and after. So we know that there are changes in the viral composition, but we don't know when it happened or how much it happened. So that's why we kind of did it in a crude way.
So they only sequenced it after the third cycle?
Yes. Well, they did before. And then so we have kind of a comparison before and after this treatment, but we don't know specifically when those changes occurred.
And you probably should have, if they did it quantitatively, you should know the fraction of the virus that's escaped versus not, to give you some calibration.
Oh, that's a good idea.
Your IW and your IM.
Right. Yeah, I guess we didn't look that closely. I think we got stuck because we only have two virus variants and they measured multiple ones, but that's a good idea. We could at least try to measure some.
Yeah, and the second thing in terms of at least downregulation of receptors and that tolerance mechanism, I mean, you could get PBMC from the animals and see how they respond in vitro to IL-15 or N803 and see if the response has been changed. So it has the downregulation that you're postulating actually has at least a measurable effect.
Yeah, so that's a good point. So they did measure the expression of IL-15 receptors in the animals that we use data for here. So we know that there's a change, but we are actually talking to them about including some functional assays in this next round of monkeys that they're infecting, so that we can get at some of these parameters, like how inhibited.
Yeah, you definitely need to do that, because there are systems in which could spare receptors. You only have to engage a small fraction of the receptors to get a reasonably full response. And so just seeing down regulation of receptor number doesn't necessarily tell you how that translates into a functional response.
Right.
Anyway, just some suggestions.
Okay, no, that's great. Thank you.
You're welcome. No, we're doing some N803 modeling also into this. So
great. Thank you so much, Alan. John
Sure.
Metzger had a question. I don't know, John, do you want to unmute yourself, or would you, if you don't unmute yourself, then I'll just ask it. Okay, he did.
Sure. Yeah, at the end of your talk, which that was really interesting. Also, I really loved your definition of systems biology. That was really helpful to me.
Okay.
So thank you. My question is, it sounded like you indicated that you could integrate multiple data sources into the model. I wrote math, but model. And I was wondering if I was hearing too much, if there's the way that you're deriving your parameters, fitting your parameters, if you're using previous parameters to inform updates, or do you mean something else with that?
Yeah, so data integration, like systems biology, could mean a lot of different things. So what we did here, since we only have the one primate data set, that's the main thing that we're focusing on in terms of calibration. However, there are some parameters that we were able to constrain and in some cases fix. I'm trying to get back here to some sort of model diagram. So if we look here, so for example, the pharmacokinetic parameters of this drug are pretty well characterized. So in this case, we took pharmacokinetic data from a completely separate study that really focused on measuring the pharmacokinetics. And we said that since it wasn't the focus of this particular study, we don't have good PK data for these monkeys, we're going to assume that the PK stuff is the same in these monkeys as in the other study. And so that's an example of where we put two different data sets together. And in this case, we just pulled the parameters from that data set. In other cases, things like, I don't know, killing rate constants or viral reproduction rates, things like that, that have been modeled in or have been captured in other models or have been measured in other assays. We can use those things to say, okay, well, most of the models that are in this area have a value between 1 and 100. So we're going to make that our bounds for our calibrator to kind of constrain the values within those biologically feasible ranges that have been identified by others.
That makes complete sense. And so it sounds like you're taking advantage of the modularity of the mathematical model, and to like where the experimentalists that you're directly collaborating with may not have data, but there's a reasonable proxy or analog in some other data set. Perfect.
Exactly. And then depending on the quality of that data, we'll either use the values. So for example, in the PK model, we know what those values are. They've been pretty well characterized, so then we'll just use them. But in other cases where we have some idea, but we're not quite sure, then we'll use those to kind of bound things, but we're not going to restrict the model too tightly because we're not quite sure what they are.
Awesome. Thank you for explaining that.
Mm-hmm.
Okay, we have two more questions. One of them's quick, so I'll just answer or ask it first. It's from Rosalba.
You can answer for me if you want to.
Oh, sorry, did I say answer? I'll just take this over and you can put my sorry. No, so the question was, why did the experimentalists consider a break of 29 days between I guess that was the second and third cycles, I think, right?
Yeah.
So maybe it's just some rationale for that.
So as far as I know, they did that because they didn't see a response in the second cycle. And so the thinking was maybe if we give them a longer break, we'll get some of it back. And I have no idea where the number 29 came from. I'm assuming it has something to do with how much it costs to keep the monkeys around at the primate center.
Yeah, I think, and I think you found that borne out in your modeling, that there was some sort of rebound basically of with that break. Okay, and then our last question is from Adrienne, which is longer again. So maybe Adrienne, do you want to ask it yourself or do you want me to ask it? Go ahead.
Yeah, I can just ask briefly. First of all, thank you so much for the talk. It was very interesting. I just wanted to know whether dosage matters, because you kind of showed that with one of those models that the drug tolerance could be the potential thing that combined with the immune regulation is what matters. Does the dose then have an impact, and would changing the space between dosages and the dose size be able to do anything?
Yeah, so the dose size is really something that I wanted to look at because that's something that I've done for other models before, but we really just don't have enough data in terms of dose response curves to be able to do that. Most of the animal models are given, I think it's 0.1 milligrams subcutaneously. So there's really only data for this one dose. And so if we did dose response, I think it would be guessing. It's possible that we could maybe go back to more of the in vitro functional assays maybe to see if there's some dose response there. We have not done that. But yeah, I agree with you that there might be a dose size as well as a dose spacing thing. We just haven't gotten to it yet.
Cool, thanks.
If you have any suggestions, let me know.
Nope.
I actually found it interesting that the IV data in the cancer study was less effective than the subcutaneous data, because I would have anticipated the other, but I guess there must be some heavy distribution and then maybe heavy elimination from the plasma. Is that right? Or
so I'm entirely making this up based on other projects and other drugs that I worked on, but just looking at the data, I would say that part of it is probably that elimination, that if it's deposited in the tissue, that it takes longer for it to get into circulation and get eliminated. So I do think that that's part of it. But I've also heard other conversations around sub-Q or IM delivery, that putting it right in the tissue where the immune cells are patrolling, where these interactions are kind of usually taking place in the tissues, it can kind of give a boost to the drug efficacy, because if it's just floating around in the blood, that's typically not where all of these stimulatory things happen. And then also maybe transporting it to the lymphoid tissue might help it get to the cells that it needs to get to.
Right. Yeah, no, that makes sense. I was thinking more of drugs, but you're right. This is like a cytokine, so that it's being picked up on immune cells. Great. Well, thank you so much for your talk. I think it obviously was very interesting, and we look forward to seeing this work progress. So I'd like to thank everyone for participating, and again, Elsje, for giving our first talk. Our next talk will be the first week of November, same time, 12 p.m. on the first Wednesday. And it will be given by one of my colleagues at the St. Justine Research Center. He works on nanopore sequencing technology, so it'll have some sort of genetic or RNA type focus. And you can find updates on our website, and I will of course send the information via email, and I will send information about this talk, which again will be posted by the Fields Institute. Thank you for that support when it's available. So thanks very much, and see you in a month.
Thanks, Morgan.